All right, as promised, I'm going to give you a short and sweet video on mutations. And what we're going to do today is talk a little bit about what a mutation is, different types of what are called point mutations, which are essentially changes kind of on the DNA level. There are also chromosomal mutations, or large chunks and pieces of chromosomes get deleted, which are much more substantial. But obviously, these can be also very dangerous as well and detrimental, or sometimes they can actually help, or sometimes they do nothing. So we're going to talk about the different types of mutations that we can see, as I said, on that DNA level, at the base pairing level. So a point mutation is a change of one base pair of a gene in like one place. Now, it can be more than just one, it can be multiple, but it's happening at the DNA level. Now, it can affect protein structure and function, obviously, because as we talked about when we talked about transcription and translation, if the original DNA is changed and mutated, you're going to change that language. You're going to change those codons, and those codons are going to code for different amino acids. Okay, so let's talk about different types of point mutations. The first one's a substitution, and this is exactly what it sounds like. You substitute one letter for another, an A for a T, or a G for an A, or something like that. There's also insertions or deletions. In this case, you're not simply changing one for another, you're actually adding or deleting a base. These tend to be much more serious because they cause what's called a frame shift. And a frame shift is when, remember that reading frame, it's every 3 amino acids, right? So like AUG, CCG, GGU. If you add one or delete one, that whole reading frame shifts and it's reading different words at that point. And I'm going to show you an example of this in a second. That is going to cause the entire protein to be different after that point, and that's very usually serious. It's going to cause a non-functional protein to be made. And then, as I said, maybe if it occurs right at the end, it's not going to be as big of a deal, but especially if it occurs in the middle or beginning, there's going to be some negative impacts of having an insertion or deletion. So here's showing you what I mean. Here's the normal gene sequence. We have our original DNA sequence at the top. We have our mRNA that has been transcribed from that DNA, and then we have the amino acids that were translated from that mRNA. It should be phenylalanine, tyrosine, glutamine, glutamine. Well, we substitute a letter, if you notice, in the third codon in the first position. We change the T or the C to a T. Well, now it codes for lysine as opposed to glutamine, right? And I want to make this point too. If it goes from one amino acid to another, there's likely going to be at least some effect. But if it has a similar characteristic to the other amino acid, like if they're both nonpolar, it may not be as substantial of a difference as if one was polar and one was nonpolar or charged or something like that. So another type, as we talked about, is an insertion and deletion. You can see both of them are causing frame shifts in opposite directions. So we have this insertion of a C here, right before the previous C in the normal one, right? So now it's CCT as opposed to CTT, and that one T gets pushed into the other reading frame, or into the next codon, and every single amino acid after that point is going to change and be different. A deletion does the same thing. In this case, we're deleting one of those A's up here. As you can see, the normal one should have an A in this position. We got rid of that. That shifts the T over, the G over, and the C over, and now we have TGC instead of ATG, and you can see again it's causing everything after that point to change. The AP exam loves frame shift mutations, and sometimes they even try, as I will try to do on your test, by the way, try to trick you into thinking it's going to be a frame shift, but on the test the frame shift is going to occur, or that the deletion or insertion is going to occur in an intron, and it won't matter because that intron is what? It's cut out, right? So it won't matter. Like mutations and introns usually don't matter unless it messes it with the ability of the spliceosome to interact and actually cut it out. So different ways mutations can have effects on the protein. One is no effect. A nucleotide, if it's in the third position, remember, because of the wobble effect, that third position one, the same amino acid is coded for by several different codons, and it's usually the third position that it can be different in, so that would have no effect. It would still cause, even though it's a different DNA sequence, it produces the same amino acid and thus the same protein. A missense mutation is when amino acid does change from a different one from the original one to a different one. And then there's a nonsense mutation, which are, again, very serious because you can just have a simple substitution, which usually doesn't have as much of an impact. As I said, a substitution can have an impact. It's just usually not as much as a frame shift does. And a nonsense mutation. So that nonsense mutation is a stop codon is coded for, it prematurely cuts the protein off because the nonsense codes for a stop codon. Stop codons, remember, add a water. A release factor comes in instead of a normal amino acid in a tRNA. The water is added, it severs that bond, and then it is free at that point. So it happens early. That can obviously have major impact on what protein is produced. So you can see different examples. Here's a no effect. Notice this original one, glycine, GGC. Well, if we change it to GGU, it still codes for glycine because of the wobble effect. No effect on the protein. A missense mutation, notice in the first, it's in the first position now, and that does change the amino acid to a serine. So that's a change in amino acid. Could have an impact. Maybe it doesn't have a huge impact. It really depends on where that amino acid is and the type of amino acid it changed to. The last is a nonsense, and you can see here you change that second codon to a stop codon. Instead of AAG, it's UAG. It stopped, and the whole thing's cut off, and you don't get a functional protein then. We can also add insertions and deletions, or we can do insertions and deletions in increments of 2 and 3 and beyond that as well. But if you ever do it in 3s or 6s, you're not going to have a frame shift because then you're just going to add an amino acid. Again, could that have an effect? Yes, but it's not a frame shift like we mentioned. And that's another thing you want to know. So you can always, in multiples of 3, it may not be as harmful. Again, it could be, right? But it probably won't be because you will not have the same frame shift if you just add an amino acid because all the ones after that will be the same. So an example of this is hemoglobin and sickle cell anemia. So here's your wild type hemoglobin. Here should be CTT, which is transcribed into GAA on the mRNA, which codes for glutamine. Now glutamine is a charged, very polar amino acid, right? Charged to the point of where it's charged. Mutant hemoglobin, as you can see here, we've changed that second position of this one amino acid to a valine. And valine is a very non-polar amino acid. And as a result, it's just the symbol or the single point mutation, the substitution that causes the sickle cell disease. Okay. Mutagens are things that tend to cause mutations. So remember, mutations happen in replication. It just happens, right? It makes a mistake 1 out of every 3 billion. So the more times the cell has to replicate itself, the more mistakes can happen, that increases your risk of mutations. And thus, as we talked about in previous units, it increases your risk of cancer, right? Mutations from actual things that cause cancer, like X-rays, UV light, certain chemicals like formaldehyde, asbestos, okay? All of those things can cause cancer. There are many other things as well. Some things, different chemicals found in cigarette smoke. Nicotine itself is a carcinogen. So those are any things that cause mutations. Now remember, if you change your mRNA sequence, right? If let's say that isn't transcribed properly, that's not as big of a deal because sometimes you'll have that and you'll make the wrong protein, but who cares because your original recipe is the same. So next time you'll make a good one, right? If you change the original recipe, though, everything after that point's a problem. So that's why mutating DNA is so much more serious than having a change in this sequence of mRNA, for example. All right, that's pretty much it. This is showing you the entire process that we just talked about, right? We have transcription here that happens in the nucleus of the eukaryote. RNA polymerase is copying that DNA sequence, that template strand, right? It's sliding along the template strand in the 5' to 3' or 3' to 5' direction, excuse me, and it's transcribing that gene in the 5' to 3', meaning it's synthesizing the new mRNA in the 5' to 3' direction. But remember that an original transcript is only pre-mRNA. It has to be modified 3 ways: 5' cap, poly-A tail, remove the introns, right? Splice them out using the spliceosomes. The poly-A tail, it helps to keep it around longer. It prevents exonucleases from destroying it, and the 5' cap stabilizes it. Then snakes its way out of the nucleus through nuclear pore. It goes to the ribosome. It finds the first start codon, right, AUG, and then it assembles itself. And then the new amino acids are brought in by tRNAs. Those tRNAs are reloaded by aminoacyl tRNA synthetases once they get their amino acid taken out. And then it keeps going until it hits stop codon. It adds a water and severs. And that is the entire process. So today we talked about mutations, different types of point mutations. Remember, frame shifts are really bad when you delete or add. Substitutions can be bad. If it's in the third position, it usually doesn't change the amino acid, though it can, depending on which one it is. And then we talked about things that cause mutations, and we reviewed the entire process. So that's your video. Enjoy the rest of your break. If you have any questions or concerns, feel free to email me as well.